Hello, welcome back to another day, another lesson. Today we're going to talk about a little bit regarding where was it? The TCA cycle, okay? So for the TCA cycle, you have a couple of carbon oxidation states first. So these carbon oxidation states change across functional groups such as alcohol, aldehyde, and carboxylic acid. And yeah, for that one, ethanol, we know that it's an alcohol with essential carbon. It's fully reduced. Acetaldehyde is an aldehyde. And acetic acid is a carboxylic acid. Again, we mini reviewed that earlier. And now we have the TCA cycle, also known as the citric acid cycle or the KREBS cycle. It's a central metabolic pathway that fully oxidizes the acetyl-CoA to CO2. Okay, so what's the purpose of the KREBS cycle? It will fully oxidize the acetyl-CoA to CO2 while also capturing energy in the form of FADH2, NADH, and GTP. How does it start? It would start with, well, the cycle would begin with acetyl-CoA, that's the 2-carbon substrate. It would condense with oxaloacetate to form citrate. Okay, so what's the beginning part? You have acetyl-CoA condensing with oxaloacetate to form citrate. After that one, you would have a couple more steps, such as like involving intermediates like isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, and malate before you regenerate the what? The oxaloacetate. So, and you have a couple of key enzymes as well, like you have the citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate, and dehydrogenases. They just to remember those, you would just need to take note of either where they came from or where they're coming from. Most likely where they're coming from. So for example, the dehydrogenases would dehydrogenate the thing it is dehydrating. So yeah, it's isocitrate dehydrogenase would be involved with the isocitrate, alpha ketoglutarase. Dehydrogenase is for alpha ketoglutarate, and etc. And throughout the cycle, high energy electrons are harvested through redox reactions that reduce NAD plus 2, NADH, and also FADH to FADH2. That's very constant. And also, even though acetyl-CoA is consumed, intermediates like the oxaloacetate could act catalytically, and that means they regenerate each turn and serve to carry and transform carbon without being depleted. Okay? And the path of carbon throughout the cycle involves the release of 2 CO2 molecules per turn, so 2 carbon dioxide. So that's where carbon dioxide came from. Because remember, you have food coming in and oxygen coming in and water going out and carbon dioxide coming out, going out. So yeah, this step would just, after the energy yielding conversion of succinyl to succinate, it would then generate GTP or ATP via substrate level phosphorylation. Okay, next. Oh yeah, we talked about this one. So you have glucose or food going inside, and O2 as well. And if you have oxygen, well, before you start talking about oxygen, glucose would be converted to pyruvic acid, right, through glycolysis. And then if there is oxygen, pyruvic acid gets converted to acetyl-CoA if you have the oxygen. If not, then you go to use the lactic acid route, But if you have oxygen, pyruvic acid gets converted into acetyl-CoA. It will donate 2 carbons into the Krebs cycle and ultimately organized into CO2. You get NADH as a result and some FADH2 as well. Just to be even more specific, so we mentioned pyruvate is produced from glycolysis. It enters the mitochondrion and is converted into acetyl-CoA. So pyruvate enters the mitochondria and then gets converted into acetyl-CoA by what? By pyruvate dehydrogenase. So who converts the pyruvate into acetyl-CoA? PDH, pyruvate dehydrogenase. That's important. Acetyl-CoA would then enter the TCA cycle. It would combine with oxaloacetate to form citrate. And what makes that? It would be the citrate synthase because it makes citrate. Okay, so pyruvic acetyl acetyl-CoA plus oxaloacetate. Since citrate synthase is the magic syrup, it would convert it to citrate. Citrate would then, after a couple of steps, regenerate oxaloacetate. And then it would produce CO2, NADH, FADH2, or NGTP. What happens to NADH and FADH2? Good question. Those would donate electrons to ETC. Where does the ETC happen? It is in the inner mitochondrial membrane. And why is that important? NADH will deliver electrons starting in complex 1, while FADH2 from succinate dehydrogenase would donate to complex 2. Those electrons would then flow to complexes 3 and 4, ultimately would reducing to H2O at complex 4. And this electron flow would drive the pumping of protons into the intermembrane space, which will establish a proton gradient. And that proton gradient would flow back into the mitochondrial membrane matrix through the ATP synthase. Why? To synthesize ATP. From what? From ADP to PI. So just to pin it down, NADH and FADH2 would donate electrons to ETC. That's happening in the inner mitochondrial membrane. And why is that important? Because the flow of electrons would drive the pumping of protons through the complexes 3 and 4, and also our ATP synthase. And there's also the part where adenine nucleotide translocator would exchange. That exchanges the newly formed ATP for ADP across the inner membrane. You also have the pyruvate carboxylase for anaplerotic OAA or oxaloacetate generation. There's also the glutamate dehydrogenase, which would convert the alpha ketoglutarate and glutamate. That would link the amino acid metabolism to the Krebs cycle. So all of them just talk. It's not just in one compartment. They talk to each other. So yes, overview. We mentioned that there's also just specific parts. Like if a TCA cycle so this is just experimental. I think like if you label, so if a TCA cycle system is incubated or labeled with carbon 14, maybe at the second position or the methyl group, you would trap the methyl carbon of acetyl-CoA as it enters the cycle. And in the first turn, you would see that the labeled methyl carbon is incorporated into the citrate, which will become one of the two CH2COO minus groups. And the thing is, due to the symmetrical nature of citrate and the rearrangement that occurs from citrate to isocitrate, you won't be able to distinguish those two. And also during the isocitrate dehydrogenase step, one CO2 is lost, but that would originate from the oxaloacetate. So yeah, that's just the carbon 14 labeling step. And what else? Okay, there's another question. There's a question here. Which pair of enzymatic steps in glycolysis carry out substrate level phosphorylation? That would be the G3P dehydrogenase and the 3-phosphoglycerate kinase. Why? Because it would involve the direct synthesis of ATP from ADP. So the G3P dehydrogenase and the 3-phosphoglycerate kinase, as mentioned, would be involved in the direct synthesis of ATP. So if you just remember this figure, then you're good to go, hopefully. There's okay. Talk about some of the like mini redox potential that would link those two. Redox is measured and compared using electrochemical cells, and it's also called the reduction potential. Generally, you have some electrodes, so you measure it under standard conditions, right? So one big concentration for the aqueous species and 1 atm pressure for the gas, 25 degrees Celsius for the temperature. And in the top right diagram, you have a cell which compares the Fe3 plus and Fe2 plus with the sum of the copper redox couples. And our copper, Cu plus, is the reductant. Fe3 plus is the oxidant, which would gain the electrons. And then you just determine the E standard or the values to compare them. And just as a reminder, the more negative E is, it means the reductant was stronger and was willing to donate more electrons. That's why it's called a reductant. It would reduce because of electrons are minus, so the sign would just become more negative. And then you have a thing called the coenzyme Q, which is a hydrophobic electron carrier which moves freely within the inner mitochondrial membrane. It will undergo reversible redox. And hello, hi Highlander, welcome back. Yeah, the coenzyme Q would just undergo reducible redox, and the fully reduced QH2 would form by accepting 2 electrons and 2 protons. Yes, and note that the electron transport chain involves a series of protein complexes and mobile transfers which carry and also transfers electrons from NADH and FADH2 through oxygen. So if you want to talk about the 4 electron carriers, complex 1 is the NADH dehydrogenase and also the NADH CoQ reductase. It receives from electrons from NADH and passes them to coenzyme Q or ubiquinone via the FMN. And then coenzyme 2, the succinate CoQ reductase, would oxidize succinate to fumarate and transfers the electrons via FAD to CoQ. So again, like just mini reviewing the 1, complex 1 and 2. 1 would receive electrons from NADH, and you pass them to coenzyme Q2 would oxidize succinate to fumarate, again succinate to fumarate, and would transfer the electrons via the FAD to COQ. And then the 3, cytochrome BC1, would transfer electrons from COQ to cytochrome C. So again, cytochrome 3 would transfer electrons from the COQ, the ubiquinol, to cytochrome C. It's very mobile because it would shuttle electrons between itself and 4. And then you have cytochrome 4, which would transfer the electrons from cytochrome C to oxygen, and that would form water. It's just the passing on of electrons. So we mentioned before that, yeah, it would generate NADH. The Krebs cycle generates NADH and FADH2 by oxidizing acetyl-CoA, which is derived from pyruvate. And these reduced electron carriers would then donate electrons to the electron transport chain, the ETC, and that's what it uses to pass stuff on. NADH would pass the electrons to complex 1, while FADH2 would give it to complex 2, and that's why FADH2 would yield a little bit less ATP overall, because it skips 1, it won't go to 2. And just they would flow until you would end up in the cytochrome 4, finally reducing oxygen to water. And now what happens in the ATP synthase, okay? The accumulation of protons would generate an electrochemical potential or proton force, and that flow is synthesized by the ATP synthase, and this will it will harness the gradient as the protons flow back into the matrix. You have its cylindrical rotor, or like a circle, a motorcycle that goes brum brum brum, and it would drive the conversion of ADP and PI, or inorganic phosphate and adenosine diphosphate, into ATP, or adenosine triphosphate. So just remember the ETC and the flow of the ATP synthase. I will sticky note that also. So it's just proton pumping and ETC. And where do all of those happen? The ETC, it happens in the inner mitochondrial membrane because it is embedded there. Next, yeah, and you also have the NADH and NAD plus shuttle. Just remember that it is the bridge that connects the energy releasing metabolism and ATP. And then you have something specific called the ANT, adenine nucleotide translocase, which facilitates the exchange of ATP and ADP across the inner mitochondrial membrane. That's why there's a difference in ATP and ADP between the matrix and cytosol. So the thing is, the electron transport chain would pump 10 protons per NADH oxidized, and of these, 3 protons would be used by the ATP synthase to generate 1 ATP, and 1 additional proton, or H+, is needed to transport the ATP out and ATP and PI in. That's why per ATP synthesized, you need about 4 protons, and essentially that would be 2.5 ATP per NADH. If you want to really be specific, it's 10 protons pumped per 10 or 4H per ATP. Just remember the 2.5. That's where the 2.5 comes from. If you really want to be super specific with how the rotor mechanism of the ATP synthase looks like, well, it would span the membrane, and you have what's that? The FO portion and the central gamma subunit. It rotates within the alpha 3 and beta 3 catalytic head, F1. That would trigger the conformational changes that would drive the ATP formation. So again, you just have the Y subunit in the middle and the alphas and betas surrounding it, and it would just undergo a conformational change and then boom, you have ATP. So yes, so far it was just a mini adventure for the tricarboxylic acid cycle, and also the what's this called, the electron transport chain. But yes, thank you for coming for that mini lecture. Bye bye, see you next time.